welcome Bart as our lecturer today. And although he will talk about Antarctica, he's here because we are working together in a project about turtles, about diatoms that live in the turtles. And as Bart is, how did you say, detective in taxonomy, you like to solve taxonomic mysteries. And we are solving some of the taxonomical mysteries of the diatoms and turtles together. But so Bart is a professor in Antwerp, and he works at the Botanical Garden as a researcher in Meise, so it's near Brussels. And he's been working for some of years now, and he published over, what we say, over 250 papers till now, and he has described more than 350 new species. So lots of them are actually from the Antarctica, and he's been working in the Sub-Antarctica and Antarctica Islands, and we are really, really looking forward to hear some of his adventures that he experienced in several of his trips. Bart, how many times were you?
9.
9, okay. So he has plenty of experience, and of course afterwards you can ask everything that you want to know, how to get there if you want.
And how much money it costs.
How much money does it cost? Okay, so Bart.
Thank you, Sunzica, for this introduction. Well, it's only 3 years that I started working on turtles and on marine diatoms, because actually I'm a freshwater biologist and actually not even a botanist. I'm a zoologist from my school's education. But since 1995, that's more than 24 years now almost, I've been working on Antarctic diatoms. And at the beginning it was the same like with all kinds of diatoms. Everybody thought that if you go there with a diatom book from Europe and you study diatoms in Antarctica, everything is clear. You can put a name on it, everybody is happy. And it's actually the same as when you go to Brazil with a bird book from Europe. And you see a bird passing and you say, oh, this looks like a gull, a seagull. Let's take my book from Europe. Oh, yes, it looks like the herring gull. It's these big gray gulls. In the Mediterranean, it's called a yellow-legged gull. And it really looks almost the same. It is the same. And everybody laughs and everybody says, this is stupid because gulls in Brazil are not the same as gulls in Europe. That's logic. But diatoms in Antarctica are the same as diatoms in Europe. That's also logic, said most people. And of course that's not. And I made my PhD in 1999, and the day after I defended with success my PhD, I realized that part of it was actually wrong. It's a very frustrating feeling, but at that time, so those that are now PhD students here in the room, it's okay to admit that you can also be wrong at the end of your PhD. The thing is, at that time, I was correct. Because I was using the books from Europe to work on the diatoms from Antarctica. And it was actually at the end of my PhD, when I defended it, that I realized that we have to work on it. And that's when I changed my career from being an ecologist to more a taxonomist and a morphologist. But for that I needed a lot of material, new samples, because there are samples from Antarctica, but then people go there and take 5 samples, 10 samples, 20 samples, and they are happy. So I went back now 9 times to different parts of the Antarctic and Sub-Antarctic region, and these are the belt of islands which are encircling around the Antarctic region. And I think I now have more than 6,000 samples in my lab. So for the next 20 years now, I can sit back, relax in front of my microscope, in front of the electron microscope, and start working on species. And more now, my students are picking up the molecular science and are upgrading my results to a higher level using the molecular sciences. Now, what I want to talk to you about today is not really about the science. There will come a very, very small part in why I go to these islands. But I want to share with you what I experienced there and how it is to do field work in Antarctica, and how it is to do field work in Antarctica in a rather primitive way. And this is the area where we will be going. This is an area called Maritime Antarctica. And how do you get there? Actually very easy. You go to the airport and you see that at 7 o'clock the flight to Antarctica leaves. That's a theory. In practice, you go to the airport, you wait there, and at 7 o'clock there's no flight. At 8 o'clock there's no flight. At 9 o'clock they say go back to your hotel, the flight will be for tomorrow. Because bad weather in Antarctica, the plane cannot either arrive there and land safely or come back from there. And the plane needs to come back immediately because it's always stormy weather. But finally one day the plane will leave. And then that's our plane. To be honest, I came today from Brussels to Zagreb in a plane much smaller than this one with propellers in a huge storm. So it was a very bumpy ride with a lot of sick people in the plane and so on. This one is also a bumpy ride, but it's a very, very, very stable plane with very, very strict safety instructions. That means that you take your own luggage and you bring it to the plane. You have to do this now in a European airport. You're shot before you end at the plane. We were in the plane actually with more people than on the passenger list. You have to do this in Europe now. I don't think you're allowed. And yeah, people were just sitting somewhere in the plane. We were attached to our seats because it was a bumpy ride. But please, be welcome. And finally we end up in Antarctica, and that's how it looks like. There is a nice tower, and there is a runway which is not asphalted, it's pure sand, so you're landing on sand, but hey, the plane runs, and the plane brought us back. And we are definitely in Antarctica. You can see it. Welcome in the base area, Antarctic base area, from Eduardo Frei, Presidente Eduardo Frei, which is one of the Chilean presidents. Because this is an island, like most parts of Antarctica, that is shared by a lot of countries. And you have there a very large chain of islands here. We start here in Punta Arenas, which is the southern, almost southern part of Chile. You can go a little bit more to the south and then you end up in Ushuaia. Last time when I was there, I took the plane from Punta Arenas and back with the ship to Ushuaia. You have to pass the Drake Passage, and to be honest, I prefer the plane, even if it's a bumpy ride, because the Drake Passage here is the most violent passage, sea passage that you can take. And if you're not seasick on that ship, you never get seasick. I was not seasick. I was happy. The very nice thing about being seasick, or about having people that are seasick, is that there's more food for the others. It's always a very good thing. So you end up with your plane here on King George Island. It's this part of the island there, but I think I have a bigger picture of it. Yep. It's there. That's King George Island, and there is an airport. And then we had to take a small boat that brought us all the way here to Livingston. And this part here is a very small ice-free area. I can show it here. It's this part here is the only part of Livingston Island where there is no permanent ice. You have snow in winter. We go in summer. That's why we go in summer. All these black dots that you cannot see on the map very clearly, but I will show them to you later. All these black dots are all small lakes. And that was what I was interested in these lakes, because in summer there's no ice on these lakes and I can take samples. But so you have the Chilean base here, and that is the Russian base. So I'm in Russia, I'm in Chile, I'm in Russia, I'm in Chile, and nobody is bothering about passports and visas and so on. So I was in Russia and Chile almost in 1 minute. There's a nice, oh sorry, go back. There's actually a nice, but you cannot see it very well, a nice Russian Orthodox church up there. We didn't go there because that was too far, and there are here some zodiacs that are waiting for us to bring us to this very large, small, rusty boat to take us one day on the Antarctic Ocean to Livingston Island. The boat is called Las Palmas. this is the bridge, we were allowed to run everywhere in the bridge, on the bridge. Yeah, it's very nice there. You can see birds and water. And that's it. And I was hoping to see a lot of icebergs. No icebergs there. We have to go much more to the south to see them. But yes, you have nice glaciers. See them here. And in front of these glaciers, from time to time, you see some nice tail coming out in the water, or actually going back into the water. And we were lucky because at one moment we were blocked for 3 hours on the ship because they had to bring some people ashore to take samples. And at that time, I think they booked them for us. But we had about 8 big whales swimming around the ship, playing and feeding. And this is the famous humpback whale, these huge things. And you can see the humps. This is his nose. On his back, he has the same things. And these things were actually feeding. So they dive very deep, and they come all up at the same moment with their mouth wide open. And you can see that they are actually taking a lot of things in them. And it's not only just small shrimps, it's also a lot of fish. And then he goes down again with a huge swipe of his tail, and then that's it. And then finally, this is Friesland. It's called Friesland. This is the highest mountain on it. It's a completely snowy mountain. But finally you see that we start to see more and more parts which are ice-free. And this is where we are supposed to be. This is the end, this is the end of the ice cap, the permanent ice cap, and all this area now is completely ice-free. And 20 years before I was there, all this here was still glacier and ice field. And we compared pictures from 20, 30, 40 years ago with what the actual situation was that we had now, and you really can see that there is an incredible melting of these ice caps. So if people say there is no climate change, I would say take another look at the evidence and you see that there is. And so we finally end up in this nice large area which is completely ice-free, and I'm going too fast. All these black dots here, all these black dots are all larger and smaller lakes that are created about 5,000–6,000 years ago. At least that is what they think, what the idea is. Because this area, this whole area here, has been ice-free for at least 5,000 years, which is very strange in Antarctica to have these ice-free areas, but they are there. You can still see here the ice cap, the Roche Dome, and this part, this dark spot here, is Clark Nunatak, which was a nunatak is a rock that is actually emerging out of the ice, and that will always be ice-free as an island surrounded in the white ice dome. But when we were there, that's Clark Nunatak, it was getting completely out of the ice. So it's no longer a nunatak. Now I think they should call it Clark's Rock, because it's no longer a nunatak. It's actually completely out of ice. And I think this was in 2009 when I was there. If I would go back this year, 10 years later, all this ice here would all have disappeared. So there is a huge problem going on this peninsula region. Now we are again in our small little Zodiac, and the Zodiac brings us about 40 minutes from the big ship, well, the 30 meters long ship, towards the shore. The reason is very simple. The ship cannot get closer because it's extremely shallow, and it has a very high difference between high tide and low tide. So it was dangerous for the ship to come closer, but with this Zodiac it was okay. We all look like Teletubbies, for those who still remember what are Teletubbies. We look like Teletubbies, very simple, because in this suit I probably will survive 1 hour in the water without 4 minutes. The water is freezing temperature, and you cool down so quickly that you need protective gear. But I can tell you I was very happy to get out of these suits because they were hanging in the machine room in the ship and they were smelling oil and grease, and you definitely don't want to know whatever smell was in there and who has been in there, but it's protection and we had to wear it. Now, this is where we are landed. To just put all our gear on the shore. That's our gear. Yeah, let's the adventure start, and that was it. The only problem is what to do now next, and we were only 8 people because we were groups of 8 people going to that island. We got immediately company, of course, because this whole beach is lying full of these meat bags, which are elephant seals, and these are the baby ones that were born this year or the year before, and they were scattered around this whole beach. But so you definitely don't want to mess up with them, because they like to cuddle, but they are about 100 to 200 kilos, and if 2 of them come on both sides, it's the end of the cuddling period. So we had to move up. We actually had to move up this hill. It's not a hill, it's actually a ridge, 5-6 meters high. But this is what we call the effect of raised beaches. The sea one day was about here, and because of the uplifting of the whole island, the whole area, you see it also in Scotland, you have a lot of these raised beaches, you had the feeling that the beach started to move. And on top of that, it was much safer, but there we set up our camp. Actually, it was set up for us because we didn't bring these guys with us. There were tents. These were our sleeping tents. We had the pink tent. I'll come to that later. We had our supply tent. That's where all the food was in. I come to that also later because it's important. We had our living room and we had our laboratory. And there we lived with 8-9 people. So again, we have our sleeping tents. Everybody was entitled to have 1 tent. We had lab and living room. And we had the pink tent with a lot of nice white pots behind the tent. The area where we are working is an ASPA area. It's called Antarctic Special Protected Area, which means these are actually areas where nobody should actually come. But for scientific reasons, you can get permits to work there. But the conditions are extremely severe. And one of the conditions is that everything that comes in has to go out. Everything. So we all had our small bottle that we were allowed to empty in the sea every day. For us boys, it was a little bit easier than for the girls, but it worked. And we all had small doggy bags. And there was, you don't see it on this picture, but yeah, no, you can see it because nobody is there. Nobody. There's a red flag sticking out. It's the same thing when you turn the wheel. You see the small red sign appear. That was also the case over there. And so after, you had to put everything in the small bag, and everybody has his own basket. And every basket, when you leave the island, goes with you. And when you finally are on the ship, and when the ship is back outside the island area, all these baskets went overboard. They are made of a very light metal, which degrades extremely rapidly. And so we expect that the dilution of the ocean will be sufficient to make sure that nothing will remain over there. That is very important because we, in our body, we are transporting a lot of bacteria, viruses, and so on. And we don't want these nasty things to be spread on animal colonies or very, very, very fragile ecosystems which you can find on these islands. I worked also on another island. It's called Amsterdam Island. It's a little bit more to the north in the Indian Ocean. But they now have, there's a huge albatross colony, and they have a very big problem with bird flu, bird cholera, avian cholera they call it, which was probably brought in with chicken or chicken meat. But now the result is that the last 5 years, not one single young albatross survived after coming out of the egg. And they're trying now to start a vaccination campaign, all because we brought in a germ that actually killed a lot of these animals. Now this is the team. We were 8 people. We had, I tried to remember all the names, not so easy. Steve was an American scientist working on fossil penguin colonies. Our Chinese friend, we have no idea what he was doing. The only thing he collected was bags with soil. But not small bags, huge bags. And he put them all around his tent. I think he was afraid his tent was going to fly away, but because none of these bags were labeled. And he wanted 250 kilos of soil to be transported on the ship and with him to China. We still have no idea what came out of these results. We have absolutely no idea. He also had to do coring in the lakes. And he had one hammer with him. So he goes into the lake, he puts the tube, he hits 5 times with the hammer, and the hammer breaks in 2. But try to find the metal part in a very ice-cold water, 1 meter deep. He had waders up to here. It was like sending somebody to a nuclear war with a pocket knife. Even if a Swiss army knife, you still will not win. So everybody tried to help him as much as possible. One day he was missing. So and it was stormy, so everybody had to go out to find him, but he was trying to carry his bags to it. So we still have no idea. But he was extremely nice. He had a huge Chinese flag with him. We all had to sign the flag. He had the time of his life. We still have no idea what his science was. Then you have Carlos and I think it's Manuel. These 2 were PhD students working on aquatic ecosystems. We had, I come to this guy later, we had a professor who was specialist in parasites, ticks, fleas living on penguins. Carlos, oh no, it's not Carlos. Manuel also. Kat Waller, that was me. Kat Waller is a scientist from British Antarctic Survey. She's English. And she was working on small copepods and lobsters and crabs and so on, living in intertidal pools. And this guy in the middle is Hilo. And Hilo was our mountain guide. He was not doing research, he was there to take care of us. He probably was our doctor. Thank God we didn't need it. He was our cook. Unfortunately, he wanted to cook. We took over after 1 day. The nice thing is, we got food from the station Juan Carlos I, which is also a little bit further on the island. There's a huge station from Spain. And all the leftovers, they froze for us in plastic bags. And we got the plastic bags, deep frozen. So what you had to do is you had to put that in hot water, because cooking, you will see later that cooking is not the easiest. You put the bag in hot water, and you melt everything. But when it's ready, it needs to be melted entirely. We usually had it melted on the outside and an ice lolly on the inside. So we took over, we skipped the plastic bags because they also delivered a lot of very nice Argentinian steaks. And they delivered a lot of tomatoes and onions. So every day we cooked tomatoes with onions and meat, or meat with onions and tomatoes, or onions, tomatoes and meat. So we had various combinations that we could make, and it was always Kat and me who took over with the cooking because we wanted something more than ice lollies. But Hilo, he was a perfect guy. If you needed help with carrying something, setting up an experiment, wonderful, he was always there. But for the cooking, I think he needs to do some more classes. So, yeah, here you can see the red flag. Huh? Somebody's occupying this. This is your house. You had a very warm and hot sleeping bag going to -20. The only thing was it was only -1 there. So at night you were, I was suffocating, it was too warm. These are some more pictures from our, this is our living room. Here we fit in with 8 people. And a very important part of it is here. This was our satellite connection to the outside world. So once per day we were allowed to send a message via internet, and that was then transferred. We could also phone, we had a satellite phone, but we decided not to use it because it was in 2009 still very expensive. And we have some protective gear here hanging. And for the rest, yeah, that's where we spent our days if we were not working. This is Hilo. Cooking, this is our famous kitchen. You see we have everything we need in this kitchen. We have even a hot stove. We could warm things up. We could have coffee. It was wonderful. And if you are a field biologist, this is why you do this. It's because you can experience something that you don't experience at home. The good thing is, after 2 weeks, you are allowed to go back home to the nice and cozy home. And this here, anybody an idea? That's our dishwasher. Yes, it's an ASPA area, so nothing dirty can come out of it. So we were not allowed to wash our plates. The only thing we were allowed to do is wipe them clean with some toilet paper and bag the toilet paper in a gray bag, and that was taken with us. Once per week, we cooked some water and we washed everything with soap decently. And every drop of water was collected in a barrel and taken with us off the island. You can also see here what the temperature is. This is the inside temperature on the 19th of January, 12.8 degrees. Outside temperature 3.9. 12.8 was almost the maximum we reached. That's inside these cabins. So we were always working and sitting there with 5 layers because it was a little bit too cold otherwise. And this is the lab. Remember, you have to fit in with 8 people, and this lab is about the size, I think, yeah, of this part here. Actually, only 2 rows. Don't mistake me that it's going to the end of the room. No, it's only the first 2 rows. That's the whole lab. You have to fit in with 8 people. We had 1 or 2 microscopes in there, 1 binocular, 1 microscope. We had a huge physical chemical lab in there. Everybody had to find his spot there. You see me here working, trying to filter my stuff. That went down in the freezer and was analyzed later when I was back in Antwerp at the university. Now this allows me to tell you 2 minutes why I do this research, and not only because I like to describe new species. That's just the collateral damage of the job. No, it's about, we want to know something about biogeography of microorganisms. And let's first talk about the distribution of macro-organisms. Big albatrosses, ferns, elephants. Let's talk about their distribution. And we know its function of ecology, its function of historical factors, and its function of something that we call accidents or coincidence. Function of ecology is very simple. The geographical distribution of a species is just the distribution of its suitable habitats. A polar bear in the Sahara is not a good idea. An elephant, and not a sea elephant, but an elephant on the ice cap is also not a good idea. So if the habitat is not suitable, the animal will not, or the plant will not live there. But there's also another thing. It's the geographical distribution formed under the influence of historical and biological factors, which means that an elephant can perfectly also live in the grasslands of Australia because the habitat is suitable for it. But the problem is the thing never got there because Australia was cut off a long time ago, and elephants at that point did not reach Australia. It's also a function of evolution and dispersion. Think about the Darwin finches, how they got dispersed over the islands and evolved during all these years. And then, of course, there is the factor of coincidence. And the geographical distribution is altered by, for instance, an extinction. A cat enters an island with small birds and eats all the small birds, and it was the only island where the bird existed. That's a possibility. It can also be the function of introductions, accidental effects. So this is what shapes the distribution of macro-organisms. But what about the microorganisms? Do they follow the same line? And now we have to go back to about 1800, where Augustin de Candolle, a Swiss scientist, made a fundamental law of biogeography. And he said the lower the organization of the body is, the more generally, the more widely it is distributed. So when you are small, you can get everywhere. And it was Laurens Baas-Becking, that's his last name, a Dutch scientist, the Dutch curator of the Leiden Botanical Garden, who in 1932 said, everything is everywhere the environment selects. And this was then later on put by two guys, actually it's three, Fenchel, Finlay, and there's a third one, Clark, but I didn't find a picture of him. Finlay and Fenchel and Finlay, they turned it into the ubiquity hypothesis. And they said that there are astronomical population sizes. Right? For those that work, for instance, with diatoms, in one drop of water, you find thousands of individuals. They have an unlimited dispersal because if the wind blows with a little bit of dust, they can get everywhere and very far and very fast. So there is no isolation of populations, which means that when there is an isolated population on an island, sooner or later, with some wind, there will come new immigrants, and your genetic isolation will be mixed up again. So you have no genetic evolution there. And so, it's what they call, there is no allopatric speciation. And there is no local extinction. Because when the population is getting very, 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 very small, suddenly there's a new group of immigrants arriving of that species that boosts up the population again, and you have again a new population. That's the ubiquity hypothesis. But this has some serious consequences. That means that there is a low global species diversity. So when we're talking about these small species, there's only a few in the world. Which means that with my European book, I can perfectly identify the species in Antarctica, because there are only a few in the world. There are no macro-ecological patterns. For instance, we know that when going south, if you go from the equator to the South Pole, the diversity in all these groups decreases rapidly. A lot of groups will disappear. So there is a trend going southwards. There is no biogeographical zonation when you have to follow their system, because everything gets everywhere. There is no separation. And of course there is no endemism, because when a species is almost on its way to become a new endemic species, he has to be set back in genetic evolution, because the normal mother population sends in some more immigrants, and everybody gets mixed up again, and you're back to zero. Now, of course, when there is one theory, there should be a second theory. And the second theory is the biogeography hypothesis. And I'd like to show this picture of the magnolias. Magnolias occur in Asia and in South America. Usually when I tell these, people raise their hands and say, I have a magnolia in my garden. Yes, you have, but it's planted. So naturally, you find magnolias here in Southeast Asia and in patches in South and North America. We had magnolias or magnolidae in the wild in Europe, but they got extinct after all our ice ages. So it was an historic event. Magnolias can grow here. They prove it because I think if I ask now who has a magnolia in his garden, that's at least half of you might raise their hands and say, yeah, we have. So the thing can grow here. We prove it. But naturally, in this part, it was wiped out. So there is an historic event that made sure they cannot survive there. Now what we think is that unicellular organisms, so the microorganisms, actually are entirely like macroorganisms. So they are function of both ecology, historical factors, and accidents. They have a well-defined geographical distribution. They show endemism, and this is the result, sorry for the M, limited dispersion. So there is something going on. But of course we have to prove this. So we have to know what is the natural distribution area of these microorganisms. And we have to check what are the parameters, both ecological and coincidence and historical. The coincidence is not always that easy to prove, but sometimes you can explain it with a volcanic eruption, and then you see that the whole island is completely barren again, and you have new pioneer species that arrive and so on. So you can prove it. So this is the reason why I work on all these Antarctic islands. Because why do I do this in Antarctica? Because the human factor in this part of the world is too large. I actually should ask all of you, when you go out, is to leave me your shoes. I will brush them and I will see what you have been carrying. Well, we have been doing this on the ship when I was going with the French expedition. We have a project called Aliens in Antarctica, and it's not about ET or Star Wars. No, it was about alien species that you find here in Europe or in South America or in North America that are transported without really wanting to transport them into Antarctica, and they survive there. So we do transport a lot. So we try not to work here to solve these questions, but we try to work here in this Antarctic region. Plus also, you have all these very, very small islands which are there. So you can test dispersal activities because the chance that something arrives there is rather limited. So this is the reason why I work on these islands. Now, for those who do not know what are diatoms, they are siliceous algae. They are unicellular, so microscopic small. They are microscopically small. I said 100 to 1/10 of a millimeter, 10 micrometers for the scientists. They are autotrophs, so they need photosynthesis, so they live in places where they have also some sunlight. And they make an external, I call it skeleton, they make an external box. And this box, the best to show you how this looks like is to take a box of cheese. You have a box, you have a cover, and you have this annoying part around it that you have to remove to get both out. And the box is not the most important because that's just the box. What is important is the cell which is lying here inside and which is surviving inside. So it's actually a box made of silica, silicium dioxide, which is actually the same as just pure glass. Now there are a lot of species. These are Swedish diatoms because I also work on Swedish diatoms. So you can see that the variability in shapes, forms, sizes is extremely large. But now, enough of the science. Let's go back to the island. This is what I saw every morning waking up in my small little cottage, in my small tent. Nice view on the beach. I could always see if it was nice weather. I can assure you we had nice weather, because otherwise you would not see pictures, because in a storm you don't go out to take pictures because you fly away with your camera. But usually it was this grayish weather that we had there. We had 2 hours of darkness because you are much more to the south, so we had about from 3 o'clock till 5 o'clock, it was actually getting dark. All the rest, it was actually quite light. Oh. You had neighbors. This is a colony of giant petrels. And giant petrels, they have a very special role on this island. It's the biggest bird. If I do this, I have almost a little bit less than one wing. They are 3 meters. This is 1 meter 80, so 1 meter 20 extra, and you have the span of this big bird. It's 3 meters. They have a very ugly outlook. They look with their big beak, with their tunnel nose here, that they use for various reasons. They detect dead animals with it. It's like their GPS because it's full of small plates inside, and with the wind speed and the wind direction, they can actually find their way back on the sea. And it's also used to desalinize their water. They drink seawater, the salt comes out here, and they have actually fresh water, nice water to drink to get inside their body. Now these are the most important animals on that island because they make sure that when something dies, it's cleaned. They're actually the vultures of Africa. These are the vultures of Antarctica. But don't forget, these are seabirds. All the birds, so all the ecological niches on that island that are normally filled in by a lot of birds are filled in by seabirds on these islands. They're extremely beautiful when they are sitting on their nest, but you can see the nice beak. The beak is about this size. Don't go too close because you lose your finger because it's razor sharp. Then let's have a look at the beach. It's full of meat bags. We already saw them. A lot of remains of algae. These meat bags are actually quite nice. You also find a lot of remains of old whaling activity because yes, we had a lot of whaling activity. I tried to lift that thing. That's about 200 kilos. I don't try this more than once because it's enormous. This was not the largest one. We had larger ones. These are these blue whale jaws. They are enormous. You find a lot of large algae. This piece is about 40 kilos. It's one of these giant kelp algae. The whole beach was littered with it. You see all these small pieces, especially after a storm, the whole beach was full of it. You find other nice, well, or not so nice remains of old whaling history. These are probably from a blue whale because these things were about this size. It's enormous in thing. If you try to lift them, it's not possible. 40, 50 kilos for one thing. It's really huge. And you find, and that was also the picture that I sent for the announcement, you find some very cozy little animals on the beach. They're about this size. They're very playful. They like to come close to you. They have a look with their nice puppy eyes, and then they open their mouths like and then it's finished with playing. You see, you're working on the water, you're working there, and then suddenly he just comes out and he sits there in front of you like trying to look. So they're not dangerous, okay? When they're older, they're less cute. This one tries to disguise him. He came up like that from the water and it was so cute with all this seaweed on his nose. I said, what the hell are you doing? This one needs to see a doctor. They have colds and he's just sniffing what I'm doing. And to show you, you can get close to them. Yes, this is not photoshopped, okay? This is a real picture. I'm just standing next to it. Now you can also see the size of these animals. They're actually not dangerous. Yes, they are, because they can do this and go back down in a fraction of a second. So you always have to keep in mind these are animals, these are wild animals, and you are trespassing in their area. So this was actually just for one picture that I come close to them, because if I wanted to show one day the people how big these things are, and this is a small one, this is a young male, this is maybe 3-4 years old. The big ones are 3 times as big and weigh 4,000 kilograms. I don't go that close to that one, okay? They can look very, very suspicious at you. And they have one big problem. Once per year, they lose their skin or their hair. and they get new. But when we lose hair, we get one hair. When they lose hair, the whole skin comes off. And you can see that. You see everywhere pieces of skin hanging. See? See? It's like they're molting. But when they mold, they're not waterproof. And a seal that is not waterproof sinks. And it's not so nice because it gets cold. So they group themselves in huge groups, about 100 to 150 animals together. And they lie there for a month. And the yellow color that you see is not the old skin. They don't move for a month. And the smell that you have there, you best compare it. You take a bucket, you fill this with shrimps, you close the bucket, you put the bucket in the sun for 2 weeks, and you open the bucket. Because I cannot bring you the smell here, everybody would go away. This is what the smell is of elephant seals. Also penguins, they have almost the same smell. It's a bit more fishy, elephant seals. But this is how it smells there. And when there is wind, there's no problem because the wind blows away the smell. When there's no wind, you don't come too close to them. But it's extremely magnificent to see that they are back because they have been hunted to almost extinction in the 30s to 60s because they used all this blubber for... No, sorry, they were extinct. They went in the 20s because they used this to put it in the oil lamps in Paris and London to light the streets. It was used fat from elephant seals, and so almost all of them died. Now I did an experiment. Why are they so big? What is in there? And I can tell you what is in there. Air. And to prove it, I took a stick, I punched it, and it went empty like a balloon. No, actually this is a mummy that was under the ice for a long time. And with the melting of the ice, you see popping up these mummified carcasses of young, this was a young one, you can see the size, but it's about maybe 1 or 2 years old, that was died, got stuck under the ice, and you see a lot of these carcasses now over there. Now there's another one also there, and that's this guy. And for this, you use your teleobjective. This is a fur seal. And fur seals have been almost extinct because people like their fur to make nice coats and hats and gloves and boots and so on. And fur seals, they run as fast as a dog, a big dog. They have very nice, long, sharp teeth as a dog. And they bite as a dog, which means infected with bacteria. And some of us have been really chastened away from the place where they were sitting because this one was coming and trying to attack. And I was on another island, on Amsterdam Island also again, and there was the ornithologist, and she saw one coming and she moved backwards and she didn't see that there was one in her back. And he took her by her leg and just snapped it. And she was in bed for 3-4 weeks with heavy antibiotics because it was very, very, very bad injury. So these things are actually dangerous. These are fur seals. This is the male, but I can tell you this is the female and she's as mean as the man. So there's no difference between sexes. It's gender equality. They're a little bit smaller, but even if they look cute, this is something we really were very careful about because they're very dangerous. They can look very, very nice, but you see these small little eyes. And then it shows that it's time to move. And then we moved. So we moved and we showed that this island is actually of volcanic origin. You have a very nice basalt formations there. And also, it's green. Yes, it's green. There's no ice because it's an ice-free area. And I think it's time to meet the forest. There is the forest. That's it. This is huge. You see these mountains, these mountains are about 2 kilometers high, and the trees are very high. I think nobody will believe me, because I took a drone and I took pictures with the drone from the forest, and you actually see here the 2 vascular plants that are growing on that island. You have Colobanthus, pearlwort, some patches of it, and Descampsia antarctica, which is a small grass species, which are growing there. This is actually everything there is on vascular plants. All the rest is mosses, lichens, cyanobacteria, and algae. And that's the whole ecosystem. So it's a very fragile ecosystem. So if I would put my foot on this, I destroy an entire ecosystem. So we were asked, it's ASPA area, to be very careful where we were walking. And we always had a huge map with all the dangerous places where not to walk, especially where the bird colonies are. And one day, we were walking on the beach, and suddenly we had about 2,000 birds flying around our heads. These are terns, sterna. Terns are extremely beautiful animals, but have the very nasty behavior that when you come too close to their nest, they aim for your eyes. We had 2,000 above us, and we had no idea why. And then I realized, because we looked at the map and there was nothing on our map. And then we looked down and we realized that somewhere them were still sitting on the ground, and I don't know if you can see them, but a lot of small stones were lying there, and on top each of these stones 1 or 2 gray brownish eggs. So we were actually, by accident, arrived in the middle of a huge stern colony, which was not on the map. So we mapped it now, and it's now nicely indicated. Do not go there. So I was leading the group of 3-4 people, and I said, okay, now we just don't look at the birds, just look at the ground and make sure you get out of this colony and that we go around it. Now, the good thing about the Antarctic tern, because this is not the Arctic tern, if this was the Arctic tern, I would now have scars everywhere, but the Antarctic tern does not attack until he has babies. So with eggs, he's quiet. They just start shouting at you and dropping things you don't like, but at least they don't attack, they don't aim for your head and for your eyes. And so we are walking on the beach. This is how the beach looks like, with a lot of mosses. All what you see here is all mosses and small lakes. And I sample all of these lakes until you reach on the beach this area here. You see the elephant seals lying down here. But this area here is one of the biggest penguin colonies of the island. And you see here the famous gentoo and chinstrap penguin. Most of them below, because the colony was divided in 2 parts, below you had chinstrap penguins. They were asked to be decent for the picture, but you know, kids, they like to play in the mud, and then yes, they end up dirty, because these are the babies. These are the adults. So they go washing, and they came out clean. Beautiful birds. Very curious. I was just sitting on the beach, and I see about 100 meters that there are some swimming penguins. They rush to the shore, and they start running to me. And then they come. Look, somebody's sitting on the beach. And then they turn around and they disappear. And then you see the next group coming. And every 5 minutes there was a group coming to watch you. And they stay about 1 meter from me. I was sitting there, so they were about a little bit small, because about this size. Very nice penguins. They also had babies, smaller babies also. Cute, huh? Look at. They have 2 chicks every nest normally. They are very, very cute. About this size. On top of the mountain, you had another species, the gentoo penguin. We call it also sometimes a donkey penguin because they produce a sound. That's exactly the sound they produce. And again, they were asked, because they had visitors, to dress up nicely. And again, they ended up dirty. So unfortunately, we have to deal with that. So these are the babies. These are the babies. These are the adults. The adults have these very beautiful white spots. And even when you are working, you always have visitors. I would dig deeper if I were you. They always come to see you. But what actually is this? This whole part here is actually an old penguin, fossil penguin colony. And we took samples of multiple layers because we know that some diatoms are typical for penguin colonies because they like the penguin shit. They like the high concentration of nutrients and phosphates and salinity. So we are going to try, I still haven't looked at the samples, but I took samples of several layers to see if we can see a shift in diatom composition following the years of occupation and non-occupation of the area. I did the same with albatross nests on Crozet Island, on another island, and there you can see that when a nest was abandoned, 3-4 years later, the flora around the nest turned back to the normal, pristine flora that was not influenced by this high amount of nutrients. So I'm going to try to see the same thing together with this Steve Emsley, the American guy, to see if we see some changes. Now, gentoo penguins are also having 2 chicks normally. They have a very special way of feeding, and I'm very happy that we humans can teach our kids to learn to eat with fork and knife, because they just stick your head in the mama's head or in the papa's head, and you just transfer all the food this way to the babies. But sometimes they are extremely cute. This is brother and sister, or sister and sister, or brother and brother. You can't see the difference. But unfortunately, and this is now not for kids' eyes, sometimes you have casualties because not everybody survives. And when that happens, there comes the cleaning ladies in the form of skuas, and they will take care of the rest. And skuas are seagulls. This is a group of seagulls, very, very, very strong birds, that if you come too close to their nest, whether it's an egg or a nest or a baby, you are attacked. And on another island, on Crozet, I ended several times flat on my belly, trying to dive when another one, as a stuka, was trying to attack me, and you sometimes really don't see them coming, and they're not hiding behind trees because there are no trees. But yes, they are extremely, extremely dangerous, extremely big, and you can see it on the bill. They have a hooked bill, even if it's a seabird. This is again a seabird, but it takes out the role of a raptor, like a buzzard or an eagle that we have in our countries. Living on the edge is not always easy, because there is a smiling assassin lying there. This is called a leopard seal. And the leopard seal is one of the top predators. We're not talking about killer whales, but the leopard seal is one of the top predators. This is a young one. It's about 2.5 meters. This was a young one. Even there, I don't come too close. Yes, I did. So he woke up. Then he smiled at me. And then he made sure that I moved backwards. And you see the nice, sharp, pointed teeth turned backwards, because if you take a penguin, you don't want to fell it out of your mouth, because you don't have hands to manipulate it. So it actually takes the penguin and moves it a few times, and then the penguin will slide down in the stomach, because the males, the big males, they can go up to 4.5 meters, which is 1.80 meters, so 3 times this. And they have a head that is about 80 to 90 centimeters. I was very happy that this was a young one. And he was really, really not happy that we disturbed him in his sunbathing. So he showed us that very clearly. This was a young one. There was also an adult one swimming around. We saw it only once when we were in the Zodiac. And the two Spanish sailors that were with us, that were supposed to pull the Zodiac on the shore, were actually not happy to go into the water, even if it was up to here, because one of these big guys was swimming around the Zodiac. And in Antarctica one day, a young biologist, Kitty, English biologist, died because she was mistaken for a seal by one of these big guys, and he took her by the head and took her 90 meters down. And when she was back up, because he realized that he didn't have a seal, but something that was a little bit chewy, because she was diving, and these tanks, I don't think he likes the metal. And she came back up, but she was already killed. She was 26 years old. That's a casualty of leopard seal. So this happens. Now let's move a little bit more back inside. This is how it looks inside. Dry, red, without vegetation. Yes, you have vegetation. When you go to the wet areas, like here the seepage areas or these small lakes, you have cyanobacteria, you have mosses, you have lichens that are growing there. But only when it's wet. On these dry areas, really nothing is growing here. And there, these are the types of lakes where I worked on. The coastal lakes, lakes close to the glacier, lakes in a higher altitude, lakes with still some snow cover. And I notice now when I have been analyzing my data that you really see different diatom groupings, communities, based on the type of lake that we have. So there is really also some separation based on ecology. And you see all these nice mats and these nice mosses and all the algae that you grow here. So there is production in the lake, even if this is the highest life form that you can find here. Yes, there is one little shrimp, this big, and that's all that is living in these lakes, except from our diatoms. And you can see this is actually the forest in the lake. This is a huge algal mat of cyanobacteria. You can have it in orange, in yellow, in green, in blue. It's all kinds of cyanobacteria, but these are the dominant life forms in these lakes. So no aquatic plants, no aquatic insects, and so on. And here you see me sampling. This is a really sample bottle. This is not the other bottle. This is a sample bottle. And this is what we do, what we fill. And then we have to walk, because everything you do there, you walk. And you preferably walk via the beach, because there the sea comes over it twice per day and it washes away your footprints, because we always had to walk on the same path. Because the footprints that I make on these islands, I can still see today where I was walking 10 years ago, because nothing moves there. It's like walking on the moon. Of course, a little bit more activity, but it's same like that. And sometimes the walk was a little bit disturbed, and you will not believe me, but we never got that boot out. We were supposed to take it out, but it was too dangerous. And yes, we are not proud of it, but this is a part that will always be on that island, because this whole area here around is extremely swampy. You can see the boot sitting there. It will always be sitting there. And I think that's what I wanted to show you. Thank you.
Questions? I think that lots of people prepared. So let's start.
You said that diatoms, you were searching for them in layers of soil, but how did they get there actually? Through penguins?
They have always been there, because you have to think it's a penguin colony 4,000 years ago. Okay. It got abandoned, and they got debris, soil, pieces of mosses, stones on top of it. So they got buried. And then there was, that's why that whole thing was actually like a domed structure. It's a small hill, even if it's only sorry? No, because we are here, the ocean is about 25, 30 meters deep, 40 meters deeper.
But aren't like diatoms ocean?
No, these are freshwater diatoms. I work on freshwater diatoms. So these are freshwater diatoms. But of course, because they have to cope with this high salinity, because penguins are marine birds, they bring water on them, they eat fish, so the excrements also contain a lot of salt. So the diatoms are the group of species that actually is able to survive in these saltier environments. But these are still freshwater diatoms. And so the the colony got buried, a new colony starts. New diatoms got buried again. So you have actually several layers of colonies. And I think Steve made the analysis. I think he found 3 or 4 colonies that have been living there the last 5,000 years. So a colony goes and comes and goes and comes. And in between there's a period, maybe 500 years of nothing. But still you have dust and stones and pieces of mosses and so on that will pile up. That's how you have different layers of diatoms. But these are freshwater diatoms. More questions?
What does ubiquity mean?
Ubiquity means that something is living everywhere, under all circumstances. It's a difference between cosmopolitan. Cosmopolitan means it can occur on all continents, for instance. Ubiquity means it has the possibility to live in all possible circumstances, everywhere in the world. That's why it's called ubiquity. Everything is everywhere.
So did you sample not turtles, but the seals?
Yes. You have to be with 2 people. Somebody starts dancing in front of the seal. The seal is looking at you, and the other one starts scraping on the back. But I can tell you that it doesn't work, because seals are hairy.
Okay.
And even if it doesn't look hairy, they have about 3, 4, 5 millimeters of hair. And you have diatoms in there, but these are diatoms that they actually drag with them when they are crawling over the soil and through a lake and so on. So I checked, and you find actually a mixture of freshwater and marine diatoms, but you don't have diatoms that are only living there. And I also washed penguin feet, but the same. Nothing is really living on them. The only thing is they are trampling, you can see that in the mud, so they have very muddy feet, but if you wash these feet afterwards, the feet are perfectly clean. The reason is also very simple. They are both living in marine and freshwater circumstances. The marine diatoms do not survive in the freshwater, and the freshwater do not survive in the marine. So they wash actually their feet without realizing every time when they go back to another habitat. So, yes.
So I didn't catch, how long did you stay?
10 days, 14 days.
14 days.
14 days. Because we were actually, this is in 2009 was the International Polar Year. And Spain had a huge project on that island, the Limnopolar, there was a sign somewhere, Limnopolar project. And they got the possibility to invite about 40 scientists from worldwide to take part of this project. But everybody was allowed to do his own work and had no connection with somebody else. And everybody got a 2 to 3 week time slot. And so you could indicate whether you want to go. And I wanted to go as late as possible in the season, because lakes with ice, for me, it's very hard to go through to get on my diatom. So I was in I think the 4th slot. There were still 2 slots coming after me. So when I left, the other group of 8 people came. And when I arrived, I saw Pete Convey and so on, they left before me. So we always changed groups. So it was a 2-week trip together with up and down there. I was about 3.5 weeks gone. Normally with the French, when I go with the French, it's 2 months, because it's 1 way of travel with the ship and 1 way on the island, 1 month on the island. So it's longer. It's boringly longer, yes. Because 1 month on a ship, it's nice, but after 7 times, we were happy that in 2012 we almost went down because we had an accident with the ship. We hit a rock. We had 27-meter gap in the ship, and that was some action. The problem was also then that all the field work was cancelled. The ship stayed afloat, otherwise I would not be there. We were all brought to the island. We played refugee camp on the island. And there's room for 40 people. We were 150. So it was crowded. But okay, it works too, but it's a pity that we lost 1 year, 1 field season. Otherwise, it would have been much nicer there.
What is your, let's say, favorite memory of all the Antarctica?
There are so many. I'm actually thinking of asking a friend of mine who is a very good, he writes very good popular science things, to maybe team up with him and make a book about all my adventures there, because after 9 times I have a lot of adventures. I think one of the nicest things that came up now was in 2007 and 2016. That was the possibility to stay for 3 nights with 4 people on a completely deserted island with only birds, plants and seals around us, knowing that whenever we wanted that there was a ship coming to pick us up. That was not a problem. But it was Simple Island. It's a totally declared nature reserve, but we were allowed, because we had the right credentials, to go there and work there for 3 days. Actually, it turned out to 4 days, because they could not pick us up that quickly, because they had work to do on the other island that took more time. And we were allowed to stay for, there's just a small cabin on the island, but we were allowed to stay there for 4 days. It was amazing. But actually, I've been there 9 times. Okay, the one with the ship accident is not so nice. In Deception Island in 2015, we were taken hostage by the Chileans, who didn't want to let us off the ship, because there were some high-ranked admirals on the ship, and they were there for a pleasure cruise. And of course, it was the army who was taking us around, so we had to follow their scheme and not our scheme. So instead of 9 days of field work, I ended up with 2.5 days of field work. That's Antarctica. But yes, it was nice to go there, but I'd leave it now. I was once taken there by my supervisor, and I'm very happy that now one of my PhD students had the possibility to go there, and that she now will take over, and I hope go there more regularly, and that I can follow from a distance from my computer and say, now go there and do that and so on. Because I have to admit that even I'm yes, and it means every time 800 meters up and 800 meters down with 25 kilos on your back, and it's for the young people now. And I have a family now, and it's always Christmas and New Year on the ship, and it's better to be home now. And I'm still happy that I can ask what I need, because I have so many people that I know now from all my trips, that I can ask, can you sample this for me, or can you bring that for me? And they perfectly do that, and they know what they take, and I don't have to go 2 months myself anymore. Because it's not that I experienced everything there, but I've been 9 times. I think I've seen the place, I've seen the penguins. They're still nice, they're still beautiful. And in a zoo, every time I see penguins, I still love them, to see them, how clumsy they are, and how mean they are, because everybody thinks, oh, they're so cute. No, they're not cute. They're mean bastards. Sorry for the word. The main thing they do every day is how to harass the neighbor for the whole day. They all sit at this distance from each other, because that's where the beaks cannot touch. I saw one who was going down because he saw something, and his neighbor comes and he hits them on the head. Come on. They have these flippers, and I'm talking about the king penguins. They hit me on my leg, and I had a bruise for a whole week, because it's a bone, it's bone hard, and they can smash you with it. Like, they're not nice. They're just mean animals. Everybody thinks, oh, happy feet, oh, nice, let's dance with the penguins. No, it's not. Okay? It's really not. Definitely not. Yes, please.
Yeah,
Yeah, sure.
for testing your just wondering, working only in Antarctica, isn't it that you have kind of fish mappings?
Yes.
Climatic
Yes. And if I would make the lecture 2 hour,
really
yes, you can.
No.
No, you're perfectly right. That's why we're not doing only Antarctica. We are combining our data sets with data sets from Canada, Sweden, North America, Africa, and so on. Yeah. But the Antarctic records, because we are preparing some big papers on only what we see in Antarctica, show that even when you only use Antarctica, it shows a lot. But we are actually we combined them with the data set from the Arctic region, from the North Pole. And when we do it in a physical-chemical way, pH, conductivity, nutrients, and so on, and you try to classify your lakes from north and from south, you can't. It's one big cloud. Which means that based on physical chemistry, on the abiotic factors in your lake, these lakes are exactly the same. Which means that if everything is everywhere and only the environment selects, the diatoms should be the same. No, they're like that. They're absolutely not. No. On these islands, on this and another island that we made a book of, we had about 210 species and 170 are endemic to that region, not to the island itself, but to that region, and not to be found outside that region. And the same applies for the Arctic. You have a lot of genera of diatoms which are extremely widespread and common in the Arctic, and I have maybe 1 or 2 species in the Antarctic. So even when you take a similar lake, a cold basin of water with low pH and low conductivity and low nutrients, you cannot say whether it comes from Norway or Greenland or from Livingston Island, the island where I was here, or another of these Antarctic islands. But if I look at the diatoms, I can immediately say, hey, that comes from there and that comes from there. So there is something going on. But I agree with you that only working on Antarctica, it doesn't work. But what we see, for instance, maybe I cannot go back to the first slide. A slide with the whole Antarctica. You have the continent, you have the islands in the sub-Antarctic islands. Yeah, it's okay. I would need another map for that. And then you have the islands with the peninsula. You have this long arm that sticks out. Yeah, you have this long arm that sticks out, and these islands, this forms an entity. And the islands which are lying here in that ocean and in the Indian Ocean, they form an entity. And if we look at these species composition there and there, then there's only 15 to 20% of shared species. And if I take the continent, which starts here, and there's a line, you cannot see this, the line is an imaginary line, it's like the Wallace line in between Indonesia and Australia. Here it's called the Gracid line. And the populations here on this side, on the continent, and the populations on the peninsula are two different worlds. So what we are now trying to prove is that even if we are talking about Antarctic region, I can immediately say, oh, this comes from there, and that comes from there, and that comes from there. So even if it's only a small sample set, we can still say it. But of course, we extrapolated it with an African data set and some European and North American data sets. So yes.
Thank you, and thank you, Bart.
You're welcome.
Also, I guess questions, time for questions afterwards. So please, a big. How long are you staying in Croatia?
We fly back on Friday.
This Friday?
Yes.
Okay. It's a pity. Anyway, we're having another great lecture on Monday. So next Monday, so less than a week, because Croatian Botanical